Okay, so thank you very much for inviting me to speak here. It's a great pleasure to be here. So I'm Emily. I'm working in Melania Krzyzewski's lab at McGill University, and I'm working for CASIN, the Canadian Aquatic Invasive Species Network. So I'll be talking to you about metabarcoding and its applications for describing biodiversity and also identifying aquatic invasive species. And if I say AIS, that's what I'm referring to. So I'll start with a quick overview of the talk. So I'll give an introduction to metabarcoding and something that's called OTU clustering, and then talk about how we've used mock zooplankton communities to validate the metabarcoding method and how we're applying what we've learned there to the analysis of a bigger data set. So CASIN sampled 16 ports around Canada with the aim to survey biodiversity and detect AIS, and then I'll wrap up with conclusions. So I don't know how many people here are aquatic biologists, but our aquatic environments are threatened by invasive species, and early detection has been recognized as great importance. So it's been shown to be easier and more cost-effective to control invading species before they become well established. So we need to have methods of detection that are capable of identifying species that are potentially at very low abundance, because invading species may first occur at low density. And traditionally, aquatic species would be identified using keys. So for example, you'd examine an individual under the microscope and use diagnostic morphological criteria. But these keys are often based on adult criteria, so it's hard to identify, for example, larval forms. So the naught layer of copepods are notoriously difficult to identify, and it's also problematic for morphologically cryptic species. In addition, it's also been shown that the sampling efforts typically employed are insufficient to identify species unless they're at medium to high density. So as I said, this could be problematic for invading species that are at low abundance. So there's been increasing interest in finding alternative methods that give us greater detection probability, and that's where metabarcoding comes in. So metabarcoding is really a way of rapidly surveying an environment. So you could go out, for example, and sample a water body. You would take a subsample of that and do DNA extraction. So you're aiming to extract everything that's there. And then what you do is you amplify an area of the genome that you're interested in, and this is your barcode region. So this is DNA-based identification, similar to traditional barcoding, but the difference here is that you're working on this very complex community. The idea is that information contained in this barcode region informs you of what species you have in your sample. So this PCR amplified community is then sent for sequencing. So we use pyrosequencing, which is just a type of high throughput sequencing. So it generates lots and lots of data. And traditionally, these data are then clustered into what's known as operational taxonomic units. And the idea is that these units, these OTUs, correspond to species. And I will explain a little bit more about that shortly. But at this point, you could just stop and you could report a number of OTUs, or if you had a database available, you could blast your OTUs against that to actually taxonomically identify them. So before I explain OTU clustering, I just want to point out why it's important that we validate this method and assess this assumption that OTUs reflect species well. So this shows data that was generated by a colleague for Hamilton Harbour. So you have a number of species plotted for two key groups, copepod and Cladocera. And in green, you have the number of species detected with traditional taxonomy, and in blue, the number with metabarcoding. So you can see that you get vastly more species with metabarcoding, and this is not unusual. Many other researchers have reported estimates of biodiversity orders of magnitude higher, so there's been lots of debate about this and trying to understand, have we been massively underestimating diversity, or is there something going on with the method? So just to kind of give you a break to address that, so OTU clustering, to explain more about how it works. So you have this PCR amplified community, so you have these barcode sequences, and then OTU clustering works on sequence divergence. So you would pick a sequence divergence threshold, and then that defines your units. So often people pick 3%. So if sequences are 3%, more than 3% different, they get pulled out into these different clusters. And what happens is that OTU clustering usually starts with the most abundant sequences, and in the end you take the consensus sequence, and that's what you would go to blast against your database. So you end up with a consensus for that whole unit. But this sequence divergence threshold often seems quite arbitrarily defined, and there's a few issues that could complicate its use. So Often researchers focus on very hypervariable regions. So we are looking at, for example, the V4 region of 18S, and this is the most hypervariable region of 18S. So it's been shown to have high levels of intragenomic, intraspecific, and interspecific variation. So potentially if you're looking at a community of very taxonomically diverse individuals, the level of variation in that marker may be quite variable. So picking a 3% divergence for everything may not be appropriate. And you can envision a couple of different scenarios happening. So, and then perhaps naive assumption, if you have 2 species, you should have 2 OTUs generated. But if these 2 species are very closely related and there's less than 3% divergence in that marker region, then the sequences produced by those species may actually join together and form a shared OTU. So if you just use OTU number, you're underestimating numbers, but also when you come to BLAST, you still may not identify the other species. On the other hand, if you have a species that has very high levels of variation in this region, you may actually find at the 3% level you generate more than 1 OTU. So you could have multiple OTUs for a species. So in this case, you would actually overestimate diversity. So the other thing to consider as well is that you have what's known as singletons in your data. So the plot I showed where you had much higher diversity with the metabarcoding, those kind of discoveries have sparked a lot of debate about the rare biosphere, with the idea that this new diversity is mostly contributed by species that are at very low abundance. And this is supported by the fact that often these species occur in your data only once. So singletons are reads that occur once across your whole data set. And it could be that if a species is genuinely really rare in your sample, you just happen to sample it once and you get 1 sequence generated. On the other hand, during PCR and sequencing, you can have errors and artifacts occurring, and given that they're random events, they'll likely just happen once, so they would also generate a singleton. So there's debate about how to treat this type of data. So now I'll move on to how we've used the the mock community method to try and address some of these issues. So I built mock zooplankton communities with set numbers of identified species. So the idea here is that you know how many individuals and species you included in the community, so when you come to do the analysis, you should know what you expect to find in terms of number of OTUs. And I included both closely related and more distant species with the idea to be able to see if we can distinguish closely related species based on our marker region. And I also included both single individuals and populations. And the idea here is that if we include multiple individuals per species, if we potentially introduce interspecific variation, does this complicate OTU clustering? So the aim was to see whether we had a correspondence between OTU number and expected species, and could we detect all species. And just to explain a little bit more about the method, we built 2 communities. So the first one had 1 individual per species, and there were 69 species in total. So I made a core community where I mixed together the individuals, DNA extracted, did the PCR, but then I also used a method where I took single individuals of different species, individually PCR amplified them, so did individual DNA extraction, PCR amplification with these tagged primers, which means that when you analyze your data, you can separate that individual out, so you can really see what sequences were generated by that individual and OTU cluster that data. And then I use that same approach for a second community, which instead of having single individuals, it had multiple individuals per species. So I had in total 76 individuals of 14 species. So I again had a core community where everything was mixed together, but then I had these populations that were separately DNA extracted and PCR amplified with the tag primers. And where possible, I had populations of different size for the same species. So again, looking at whether if we introduce multiple individuals, what effect does that have? So we analysed the data using UPass, which is a relatively recently developed algorithm, and the default threshold that it uses is 3%, so we started by analysing the data that way. So just to summarise what we had, so we had single individuals of 72 different species and populations of 13 species, and we found that between 33 to 84% of those individuals or populations generated a single OTU. So you could argue in the majority of cases there was a good correspondence, but in some cases we had to use more than 10% divergence threshold to get a single OTU. And we also found that in general, including singletons in the analysis, so those sequences that occur once, resulted in more OTUs per species, so generally complicated the correlation. And then when we had those multiple individuals per species, the results were less clear. So we would have cases where multiple OTUs were generated for populations, but there wasn't a clear pattern that when you had more individuals, you got more OTUs that seemed more sporadic. So not necessarily clear pattern. And we did also find that we couldn't distinguish closely related species. So for example, for the genera Artemia, Daphnia, and Gammarus, we had closely related species and we weren't able to distinguish those in the data. So to summarize that, there's a few things going on. So first of all, this highlights how it's important to consider the amount of resolution in the marker, the barcode region that you use. So as I said, for Daphnia, we had a couple of closely related species, and we found that they were joining together to form a single OTU. And this was also the case for some Balanus species, so you can see that if you have less than 3% divergence, so if you can take the reference data sequence from NCBI, for example, for your species, and if you see that you have less than 3% divergence, then using a 3% divergence threshold, you're not going to be able to distinguish them, so you have this one OTU forming. On the other hand, this also shows the importance of having reference databases, so with Corbicula, for example, the Asian clam, we had multiple OTUs being generated for a single individual or for multiple individuals. And if we just stopped with OTU number and didn't have the tagging method, we might interpret these as different species. But because we can blast them against a database, we can see that they're all matching Corbicula. And as you can see, as I said, the pattern, the number of OTUs generated by the populations of different size, there's not an obvious trend there. So this could be more just like sampling if these are genuine alleles, just random sampling across populations if there is intergenomic variation. We also found that although singletons often resulted in more OTUs per species, sometimes they did allow the identification of what could be rare species. So Brachionis rotifer, when we analysed the data without singletons, it didn't generate an OTU. But when we did, we had an OTU form that was formed of 4 singleton sequences. So this individual was at very low abundance, and when we had the singletons, we could detect it. So this potentially lends support to the argument that singletons could be genuine sequences when you're analysing natural data. So all of this has really led us to think that maybe what we should be doing is just skipping OTU clustering. So OTU clustering is useful when you don't have taxonomic information available because you're still able to describe your community in some way. So for example, if you're analysing a bacterial community that has not been described in any way at all, then you have to use some kind of, so basically you're just describing genetic diversity in the sample. But in cases where, for example, if you're detecting aquatic invasive species and you have a target list of species, if you make sure that you have reference sequences available for those, then why not just blast your reads against those instead of using OTU clustering? So now I'm going to talk about how we've gone on to analyse a bigger natural port data set. So as I said, KSIN sampled 16 ports around Canada, so 4 ports in 4 different geographic regions. So we have Pacific, Arctic, Atlantic, and the Great Lakes. And all those different ports generated over 7 million sequences. And if we just blast those without OTU clustering against an 18S database, we find that we can taxonomically classify to the family level 95% of those reads. And this is just this is actually a figure produced by a colleague who I'm working with, but it's just really to show how you are able to, without OTU clustering, you can get at least to the family level estimation of diversity. And you can see that our sample is mostly composed of crustaceans. And then just to show as well how we get patterns when we look at the different geographic regions, we have patterns that kind of make sense in that the 4 ports within a region show similar patterns of diversity. So I know these figures look kind of complicated, but the different colours represent different genera, so just showing how in the different regions you get different patterns in terms of diversity coming out. And then moving on to detecting aquatic invasive species. So the way we did that was we compiled a list of invaders from resources, online resources in the literature, and I checked to make sure that those species had 18S sequences available in databases. And then we blasted those reads that were generated with the ports against the reference database and picked out the invaders. So we found 23 AIS, and when I say that they're unambiguously detected, that means that because there were some cases where a single read would blast against multiple species with equivalent blast score. So based on the blast result, you can't distinguish them. So the 23 AIS were cases where those reads blasted uniquely against that species. So there are cases where we cannot confidently say for sure that we've detected an AIS because it could be a closely related species. So for example, for Maya arenaria, the soft-shell clam, there's reads that also blast against 2 other species equally well. We also tested OTU clustering just to see how the results would differ, and we found that when we OTU clustered, 9 of those AIS were no longer detected, and for a number of those species, I looked at the reads and mapped them back to which OTU they joined, and I found that they were joining OTUs, forming OTUs with closely related species. And just to kind of explain more how this is a problem, so for example, if you have, Maya arenaria reads, and then you have reads of closely related species that could be more abundant. So I said that OTU clustering generally starts with the most abundant reads. So Maya arenaria might join this OTU, but because it's not the most abundant, when you make your consensus sequence, another closely related species will be represented by that consensus. So when you blast it, you'll detect that, and this masks the presence of Maya arenaria. On the other hand, you could have scenarios, like I said, where even when you're blasting reads, based on the blast results, you cannot distinguish them. So this is a case where your barcode is not giving you enough resolution in that group. And I know these phylogenies trees don't look particularly nice. The colors are kind of funny, but this is just to show that in some cases, taking a phylogenetic approach can help you to be more certain about what species you have. So in this case, Cercopagus is a species that's closely related to Bithotrethes, so more than 98% similar, but when you make this tree, you can see that the Bithotrethes sequences do separate out. So looking at this, you feel more confident that the reads you have do actually fit with Cercopagus. But then you can have other scenarios where, for example, Castus minus is the European green crab. Based on this, so if you include reference sequences for closely related species, the Japanese crab, Atlantic blue crab, it's more of a mess. It's much harder to see. Your reads are also clustering closer to those species. So we found that particularly for crab species, our marker is very difficult to distinguish different species. So again, if you were interested in this species, you'd have to potentially think about a different region. So now just to conclude everything. So we've seen these 2 different problems, the collapsing and oversplitting issues. So both of these could result in miscalculation of diversity, so either over or underestimation of diversity. And this could be particularly problematic for detection of aquatic invasive species when you really want to be sure which species you're identifying, because you don't want to report an invasive species somewhere when it's not really there, but at the same time, you don't want to miss it. So also this all highlights the importance of growing these well-developed molecular databases. So it's really important to have reference sequences to be able to blast your OTUs or reads against. So there needs to be a really concerted effort to make sure that we have well-developed databases. And also when you're designing these kind of experiments, these kind of surveys, really thinking about the barcode region that you're using and how much resolution you need in different groups. And potentially so the benefit of something like 18S, for example, is that it's quite easy to get this to amplify across many different groups. So CO1 has been used lots in barcoding, but it's problematic to get it to amplify across very diverse phylogenetic groups. So potentially you could envision using this broad approach where you use a broadly amplifying primer, and if you get, for example, Castus minus coming up, you could use a more targeted primer for that group to really confirm that you have that species. And in our lab, there is a master's student who is working on developing primers for CO1, so potentially using more of a cocktail approach where we have CO1 primers specific for different groups. So I'll finish there by saying thank you to my supervisors and collaborators and people that contributed samples for those mock communities, and of course to McGill and NSERC and Kaysen, and again for inviting me today. Thank you.
Does anyone have any questions for Emily? Thanks, Emily. Are you interested in abundance, or trying to work out the abundance from your barcode?
Ideally, we would like to, but I think it's very difficult to do that. So you could use read depth, or number of reads, as some estimation of abundance, but I think there's so many different things that could affect that, so primer, biases, and it doesn't necessarily correspond to actual biomass. But it would be really interesting to try and investigate that further.
The big one is copy number variation on your ribosomal gene, because that can vary quite wildly between even closely related, even within a species.
Yeah, yeah.
So a second question for no one. When you said that you had identical blast hits, what was that based on? Is that an value? Or?
Yeah, we use score, actually. So we filtered our blast outputs by using a minimum overlap of like 97%. Sorry, minimum percentage identity of 97, and then the length we also filtered by minimum length, but then when it came to the actual distinguishing top hits, we based that on score.
Because one thing you could do is, because you have a database of the sequences, you could try and work out what are characteristic SNPs in there and use those to try and guide those more difficult decisions.
Yeah, and that I guess links to the kind of phylogenetic approach, how then when you look more in detail at the sequences, how you can then see differences. So yeah, that's a good point.
Any other questions? All right, well, thank Emily again.